Distinct tumor microenvironment derived from aggregated genomic data. Suggestions for personalized treatment strategies for cutaneous melanoma. Hello, and welcome to my seminar. My name is Michael Balas, and I'm very excited to share some of the research our team has been doing in the Division of Neurosurgery, affiliated with the University of Toronto. Let's first start with a brief background on immune checkpoint inhibitors. The primary tumor is the first place where cancer cells proliferate in tissues. The growth of this tumor is sustained by a diverse group of cells, all interacting with one another, something we call the tumor microenvironment, or TME. The TME consists of neoplastic cells, in addition to stromal and immune elements, all of which contribute to sustaining the tumor. It's been a universal observation across different neoplasia that interactions between TME components are important in tumor formation, maintenance, progression, and determining treatment response. For example, the exclusion or dysfunction of T lymphocytes in the TME is a key part of the oncological process of immune evasion for tumor cells. Modulation of this mechanism by immune checkpoint inhibitors, or ICIs, has been the new therapeutic frontier for solid tumors like colorectal cancer, renal cell carcinoma, non-small cell lung tumors, and melanoma, which is the focus of this talk. Melanoma is a form of skin cancer that originates from melanocytes, melanin-producing cells in the skin that are responsible for your skin's color and pigment. Now, the efficacy of these ICI treatments is highly variable, with many determinants of success including immune cell abundance, neoantigen load, antigen-presenting defects, or even intestinal microbiota. Furthermore, gene expression signatures of T cell and tumor bulk have also been identified to predict treatment response to ICIs. With the recent and rapid evolution of data analytics, we are now able to identify and characterize disease subgroups that share molecular and clinical features, thereby representing the next frontier in diagnostic, treatment selection, and prognostics. In this study, we hypothesized that there are clinically homogeneous subgroups with shared molecular features for cutaneous melanoma, the most malignant skin tumor. We sought to see whether they exist, identify them, and eventually promote personalized treatment programs based on patient stratification. So how did we go about testing this? Through the aggregation of multimodal genomic data. To test our hypothesis, we first collected open source, freely available data from the Cancer Genome Atlas, or TCGA. We identified 471 specimens with the histological diagnosis of cutaneous melanoma. Normalized gene expression and methylation profiles were extracted, and all samples were annotated with mortality and metastatic status. So, what did we do with this gene expression and methylation data? We combined them together, and we did this using the technique of similarity network fusion. You can learn more about this in the reference at the bottom of the slide, but briefly, it works by converting the original data matrices to similarity matrices of individual patients, which are subsequently used to create similarity networks. The transcriptomic and methylation networks are then fused through an iterative process which involves combining the sparsified matrix of one data modality with the original similarity matrix of the other. This process creates two status matrices, one for transcriptomics and one for the methylation data, which are then averaged in the end of the iterative process to create an overall status matrix, or fused patient similarity network, which will integrate the similarities for both datasets. Later, we'll subject this fused matrix to clustering and outcome predictions. To estimate the immune cell components of each tumor, we deconvolved bulk gene expression data using a well-established technique called CyberSort. First, we obtained a signature matrix verified by the creators of CyberSort that has a series of markers assigned to each immune cell type. This signature matrix is then incorporated with our bulk tumor data into new support vector regression to derive cell fractions and their proportions, as seen on the right. To assess immune cell dysfunction, we analyzed the enrichment of immunological signatures from the Molecular Signatures Database, or MSIG-DB, which can be found within the Gene Set Enrichment Analysis, or GSEA, online resource. Again, this tool has been described elsewhere, but briefly, it's a computational method that determines whether a predefined set of genes demonstrate statistically significant, concordant differences between phenotypes, which in our case are tumor microenvironments. The primary result of GSEA is the enrichment score at the top, which reflects the correlation of the gene with the phenotype. Finally, we investigated interactions between different cell types in the tumor microenvironment. On the left of the slide, you can see we correlated the expression of individual genes across the 471 melanoma samples with the average expression of markers specific to CD8-positive T-cell treatment response and cancer-associated fibroblasts. We then selected the group of genes that positively correlated with both cell fractions, defined using cutoffs of an R-squared value greater than 0.4 and p-value less than 0.001. We consider these candidate genes as positive regulators. On the right of the slide, we isolated candidate ligand receptor pairs that drive different microenvironment phenotypes for further study. To achieve this, we utilized multicellular networks within the functional annotation of the mammalian genome catalog, or PHANTOM. The previous gene list derived from the correlation analysis was used for this. After applying the techniques I just described, we discovered distinct tumor microenvironment clusters of melanoma. Okay, so we ran our similarity network fusion and got our combined status matrix, there on the left. Next, we performed spectral clustering on the matrix and identified two subgroups, one with 325 patients and the other with 146. Cluster membership was primarily determined by tumor methylation profile. Now, after analyzing the composition of the tumor microenvironment in each cluster, we found that the larger group had high purity, low immune, and low stromal fractions, which we call the high purity group. The smaller group had low purity, high immune, and high stromal fractions, which we call the stroma-rich group. These distinctions are highlighted in the box plots to the right, which demonstrate significant differences in each of these scores between both groups. The lower proportion of immune cells in the high purity group suggests that tumor cells in this kind of microenvironment evade immune responses through T-cell exclusion. This is in contrast to the stroma-rich group, which we consider to be dominated by immune cell dysfunction. To test this hypothesis, we further deconvolved the immune cell composition of each cluster and characterized gene expression programs associated with T-cell dysfunction. After estimating the immune cell fractions of each cluster using CyberSort, we discovered significantly more CD8-positive lymphocytes, M2 macrophages, monocytes, and regulatory T cells in the stroma-rich group compared to the high-purity group. This is shown in the plot to the left. Further GSEA analysis demonstrated significant enrichment in T-cell exhaustion signatures in the gene expression profile of the stroma-rich group. These findings are in keeping with distinct mechanisms for immune evasion for each cluster. An immune-suppressed microenvironment with T-cell dysfunction in the stroma-rich group, and immune cell exclusion in the microenvironment of the high-purity group. Thus far, we discovered a distinct, homogeneous cluster of melanoma microenvironments with low purity, high stromal, and high immune fractions with demonstrated dysfunction. The next step was investigating the biological processes driving the interactions between cells in this microenvironment. To explore this, we correlated all observed gene expression values with the abundance of cancer-associated fibroblast fractions and CD8-positive treatment response programs to PD-L1 immune checkpoint inhibition. The candidate genes that exceed our specified correlation thresholds are highlighted in red on the left scatter plot. This analysis is built on the assumption that genes positively correlated with both cancer-associated fibroblasts and CD8-positive cell signatures of treatment response are candidates for mechanisms of cell-to-cell interactions. From this correlation, we extracted all of the candidate-positive regulator genes for fibroblast CD8-positive response programs, the red dots, and these all annotated to relevant ontological mechanisms such as antigen presentation. To further test these interactions, we analyzed the effects of receptor-ligand pairs between fibroblasts and CD8-positive cells using the PHANTOM online repository. In keeping with our annotation findings, we detected an HLA-A and CD3D interaction between cancer-associated fibroblasts and CD8-positive lymphocytes. So, where do we go from here? Well, first, to summarize, we've identified two distinct homogeneous states in melanoma microenvironments. One high purity state with low immune and stromal fractions, and one stroma-rich state with low purity and high immune fractions. Furthermore, we found that the stroma-rich phenotype carries several features of a suppressed and dysfunctional immune microenvironment. We also found that the high-purity phenotype is suggestive of poor response to immune checkpoint inhibition, indicated by its ability to evade T cells. Ultimately, these results may be translatable into early stratification of melanoma treatment response based on methylation profile and enable personalized implementation of immune checkpoint inhibition. Going forward, we want to verify these findings in histological specimens, as well as verify them with single-cell analysis. And this is important, because right now, we're only looking at bulk genomic data and inferring cellular characteristics and interactions. To give an analogy, currently, we're looking at and tasting a juice smoothie, but we're also trying to guess what the ingredients are. Despite our best efforts, we can never be sure what they are until we examine each fruit, each component, distinctly. In doing so, we can combine the right ingredients, in the right combinations, and in the right proportions until we perfect the recipe. Thank you for listening to the seminar. If you'd like to reach out, please feel free to email me at the address on the screen. All the best to you.